Today I am going to explain about topic of nucleic acids. So nucleic acids are largest macromolecules. So largest macromolecules. So nucleic acids are largest macromolecules. These are present in acid insoluble fraction. Acid insoluble fraction. So molecular weight more than 10,000 Daltons are present. So nucleic acids are largest macromolecules. These are present in acid insoluble fraction along with proteins, carbohydrates and lipids. So molecular weight approximately 10,000 Daltons. These nucleic acids are heteropolymers. Nucleic acids are heteropolymers. That means monomer units. Different types of monomer units are present. So these are different types of monomer units are nucleotides. So nucleic acids are heteropolymers. These are constituted by or composed by different types of nucleotides. So nucleic acids are polymer of nucleotides. Different types of more number of millions of nucleotides are present in nucleic acids. So nucleotides are a building block of nucleic acids. So four different types of nucleotides are present in nucleic acids in both DNA and RNA. Their nucleic acids were first discovered by Frederick Mitchell, scientist. So nucleic acids are first discovered by Frederick Mitchell, scientist, in nucleus of first cells. WBC, nucleus of first cells. He called it nuclein. So nucleic acids are first discovered by Frederick Mitchell, scientist, in a nucleus of first cells. WBC. He called it nuclein. Then Altman scientist in the year of 1889, the year of 1869. Altman scientist called this nuclein as nucleic acid due to acidic nature of this nucleic acids by using basic dyes. So nucleic acids were first discovered by Frederick Mitchell, scientist, in a nucleus of first cells. He was called it nuclein. Later Altman scientist was called this as a nucleic acid due to acidic nature of these chemical substances by used basic dyes. Where nucleic acids are genetic material in living organisms. Nucleic acids are genetic material in all types of living organisms, including virus. Then these nucleic acids are composed by three components. So three main constituents are present. So these are first one nitrogen bases, second one pentose sugar, third one phosphoric acid, phosphate moiety. So nucleic acids are composed by three main components. So these are nitrogen bases, pentose sugar, phosphoric acid. So first one coming to nitrogen bases. So these nitrogen bases are heterocyclic organic compounds. So these nitrogen bases are heterocyclic organic compounds. These contains nitrogen in the ring structure of nitrogen bases. So nitrogen bases are basically two types. So these are purines and pyrimidines. Purines and pyrimidines. So first one coming to purines. So purines are two types. So these are adenine and guanine. Purines are two, adenine and guanine. So first one adenine. So adenine, this also called as a 6-amino urine. So formula is a C5H5N5. So purine also called as a 6-amino urine. So formula is a C5H5N5. So these are the 9-membered structure. So 9-membered structure. So double rings are present. Double ring. Their nitrogen positions are 1. So nitrogen positions are 1, 3, 7 and 9. So nitrogen positions are 1, 3, 7 and 9. So remaining are carbon positions. So why it is called as a 6-amino urine? So why? Because nitrogen amino group occupied at 6th membered position. So 6th carbon position. That's why it is called as a 6-amino urine. So adenine also called 6-amino urine, C5H5 and N5. So it is a 9-membered structure. Double ring is present. Nitrogen positions are 1, 3, 7, 9. So next one, guanine. So guanine also called as a 2-amino 6-oxyurine. So C5H5N5. Whoa. So it is also 9-membered structure. So double ring is present. Nitrogen positions are 1, 3, 7, 9. So this also called as 2-amino 6-oxyurine. So at the second carbon position, amino group is present. Next one, at the 6th carbon position, oxygen atom. So guanine. So purines are two types, adenine and guanine. These purines are 9-membered structure. Double ring is present. Nitrogen positions are 1, 3, 7, 9. Next one coming to pyrimidines. So pyrimidines are 3. So these are cytosine, then thymine and uracil. So first one coming to cytosine. So generally purines are 9-membered double ring structure is present, but pyrimidines are 6-membered single ring of structures. So cytosine formula C4H5N3O. It is also called as a So next one coming to pyrimidanes. So pyrimidanes are generally three. These are cytosine, thymine and uracil. So these pyrimidanes are a six-membered single ring of structure. Nitrogen positions are first and third. So cytosine also called as a 2-oxy-4-amino pyrimidine. So why? Because amino group located positioned occupied at fourth carbon position, at the second carbon position, oxygen atom. Then thymine also called as a 5-methyl 2,4 dioxypyrimidine. That means methyl group CH3 group occupied at the fifth carbon position. Then uracil. So uracil also called as a 2,4 dioxypyrimidine. Dioxypyrimidine. Then in DNA purines adenine and guanine. Then pyrimidines cytosine and thymine is present, but in RNA instead of thymine uracil is present. So purines adenine and guanine are present in both DNA and RNA, but in pyrimidines in DNA thymine only present, then uracil is present in RNA only. So in DNA 2 purines adenine, guanine, then 2 pyrimidines cytosine and thymine is present. In RNA 2 purines adenine, guanine, then pyrimidines are cytosine and uracil is present. So nitrogen bases. So next one coming to sugar, pentose sugar. So sugar, pentose sugar. So it is a 5-carbon simple sugar, monosaccharide. This sugar is a 2 types, deoxyribose sugar and ribose sugar. Pentose sugar, 5-carbon sugar. So it is a 2 types of deoxyribose sugar and ribose sugar. So deoxyribose sugar present in DNA, ribose sugar in RNA. The main difference between deoxyribose sugar and ribose sugar, at the second carbon position in deoxyribose sugar, lack of oxygen atom, that's why it is known as a deoxyribose sugar. In ribose sugar at second carbon position OH group is present. So formula is a C5H10O4 C5H10O5. So due to this difference, DNA is known as a deoxyribonucleic acid, or RNA is known as a ribonucleic acid. So next one, phosphoric acid, phosphate moiety. Phosphoric acid, phosphate moiety, H3PO4. So formula is H3PO4. 3 reactive OH groups are present. So in these 3 reactive OH groups, 2 OH groups form the sugar and phosphate backbone of nucleic acid. So this phosphoric acid gives to acidic nature of nucleic acids. Next one, nucleoside. So nucleoside means the nitrogen base joined with pentose sugar. So nitrogen base linked with pentose sugar through the N-glycosidic bond is known as a nucleoside. So dehydration by the elimination of water molecule. Any nitrogen base, purine or pyrimidine. So purines adenine or guanine. So linked with pentose sugar by the elimination of water molecules, N-glycosidic bond through the N-glycosidic bond. So first carbon position of pentose sugar, they are linked with the 9th nitrogen position of adenine or guanine. That means purines. So first carbon and 9th nitrogen position is known as a nucleoside. In pyrimidine, first carbon of pentose sugar, then first nitrogen position of pyrimidine. Cytosine, thymine. In RNA, uracil. So nucleoside. So by this linking through the N-glycosidic bonds from the 4 nucleosides. So it is a adenosine, vanosine, then cytidine and thymidine. 4 types of nucleosides. Next one, nucleotide. So nucleotide is phosphoric acid. That means a nucleoside joins with the phosphoric acid. That means phosphate moiety linked with the nucleoside through the ester bonds. This is known as a nucleotide. So phosphate moiety link with the nucleoside. So for example, adenosine is a one nucleoside. So phosphate moiety, phosphoric acid joins with the nucleoside adenosine from the adenosine monophosphate through the ester bonds, hydrogen of phosphorus, then HU of this pentose sugar at the 5th carbon position. So adenosine monophosphate, then by the joining second phosphate and third phosphate, so alpha, beta and gamma, 3 inorganic phosphates from the ATP, adenosine triphosphate. Nucleotides, so like this vanosine, then linked with the phosphate moiety from the nucleotide. That means deoxyadenylic acid in DNA, in RNA adenylic acid, then deoxyvanillic acid, then vanillic acid in RNA. Next one, pyrimidines, cytidine and thymidine. By linking here from the cytidylic acid, deoxycytidylic acid, then deoxythymidylic acid. In RNA, cytidylic acid and uridylic acid. So 4 types of nucleotides. So nucleotide is nothing but the nitrogen base, sugar and phosphoric acid. So phosphoric acid joins with this nucleoside through the ester bonds by the elimination of water molecule. So generally phosphoric acid, phosphate moiety joins with the pentose sugar at the 3rd carbon position with the OH group in sugar. Same phosphate moiety join with the neighboring sugar at the 5th carbon position by 2 ester bonds, elimination of 2 water molecules. This is known as a phosphodiester bonds. Phosphodiester means 2 ester bonds. Same phosphate moiety, phosphoric acid joins with the 2 neighboring pentose sugars at the 3rd carbon position and the 5th carbon position. So here at the 3rd carbon position, 3 OH group is present. This end is known as a 3' end. In same stand at another end at the 5th carbon position, 3 phosphate moiety is present. This is known as a 5' end. 5' end and 3' end. In another strand of the DNA in reversible in a condition. So 5' end to 3' end. So in this another strand in this end at the 5th carbon position, 3 phosphate moiety is present. This is called 5' end. In same strand in another end at the 3rd carbon position, 3 OH group is present. This is called so 3' end and 5' end. So polarity, so nucleoside and nucleotide. So nucleic acids are generally 2 types. So these are DNA and RNA. So DNA means deoxyribonucleic acid. RNA means ribonucleic acid. So first one coming to DNA, deoxyribonucleic acid. So DNA macromolecule, largest macromolecule. So DNA is a polymer of nucleotides. So four types of nucleotides are present. These nucleotides are building block of DNA, both DNA and RNA. So millions of nucleotides are present in DNA. So for example, in human beings, so approximately 6.6 into 10 to the power of 9 base pairs are present. In prokaryotes, so Escherichia coli 4.6 into 10 to the power of 6 base pairs. One base pair is nothing but two nucleotides. So complementary nitrogen bases. Then DNA act as a genetic material in majority of organisms except some plant and animal viruses. Then DNA generally double standard condition. Mostly DNA double standard condition except some bacteriophages. So coliphase bacteriophage 5 into 174 single standard condition. Then bacteriophage M30 single standard condition. In rest of the majority of organisms, DNA is a double standard condition. Then DNA located in prokaryotes in nuclei and plasmids. Nuclei and plasmids. So freely in cytoplasm of a cell. So DNA located in prokaryotes in nuclei and plasmids. So plasmid is an extra chromosomal DNA. So these are the single chromosomic condition. Single chromosomic double standard DNA freely in cytoplasm. Histone proteins are absent. There in eukaryotes, so this DNA located in a nucleus. Located in a nucleus as a chromatin. At the time of cell division, this chromatin condenses to form a chromosomes. The number of the chromosomes is a constant and particular species. So in eukaryotes, DNA located present in a nucleus, well, because well defined nucleus is present in eukaryotic members. Then in eukaryotes, DNA also located in cell organelles, mitochondria, then chloroplast. So this is known as a organelle DNA. In eukaryotes, DNA also present in mitochondria and chloroplast. This is known as a organelle DNA. Next one. So DNA carry the genetic information, heredity material from one generation to another generation. This heredity information coded in nitrogen bases. Next one, DNA. So DNA carry the genetic information, heredity information from one generation to another generation. This heredity material coded in nitrogen bases. There DNA also permits, DNA has the property of replication. DNA has the property of replication. That means DNA to DNA. This is known as a autocatalysis. Autocatalysis. So autocatalysis is a nothing but of DNA act as a catalyst to formation DNA from DNA to DNA. So DNA has the property of replication, autocatalysis nature. By the replication, DNA form the duplicated daughter DNAs. Then DNA also gives rise to RNA. then proteins, enzymes and other biochemicals except other than DNA. This is known as a heterocatalysis. Heterocatalysis, autocatalysis and heterocatalysis. Then crossing over also takes place while the cell division in prophase 1 in Pachytena stage. This crossing over permits to recombinants, that means variations of characters crossing over. So crossing over permits to recombinants. So crossing over will helps to formation of recombinants. Next one, by the changes in nucleotides or one nucleotide nitrogen base is replaced by other nitrogen bases. These lead to mutations. So mutations will be useful for formation of new plants and animals, new plants and animals. So mutations. Then in organism's body, different body parts are present. So these body parts differentiate from each other due to functioning of specific strands of DNA or specific parts of DNA. So in organism's body, different parts are present. These parts are differentiated due to functioning of different structures of DNA. Then in life cycle of organism, different growth stages are present. These are different growth stages also due to internal clock function of DNA. Internal clock function of DNA. Then double stranded DNA proposed by Watson and Crick. Watson and Crick proposed a double helical structure of DNA in the year of 1953. So double helical structure of DNA proposed by Watson and Crick. So based on X-ray diffraction. X-ray diffraction studies of Franklin and Wilkins. Franklin and Wilkins. So Watson Crick in the year of 1953 proposed the double helical structure of DNA based on the X-ray diffraction of Franklin and Wilkins. So Watson Crick along with Franklin were awarded Nobel Prize in the year of 1962. So it is a hallmark of proportion by Watson and Crick, double helical structure. So these double helical structures show the secondary structures of nucleic acids, especially DNA. So types of DNAs. Types of DNA. So these are A type, B type, C type, D type and Z types of DNAs. So generally DNAs are right-handed in a manner. So A DNA is a right-handed, B DNA is a right-handed, then Z DNA is a left-handed manner. So it is a right-handed manner. Right-handed helix and left-handed helix. So A, B, C, D is a right-handed manner, Z is a left-handed manner. So DNA generally spirally coiled condition. So this entire helix is a intertwined or spirally like a ropes like a condition. So due to this coiling, number of turns, number of spirals, number of coils are present. So in B DNA, number of bits. So 10 base pairs, 11 base pairs, 10 base pairs, 9 base pairs, 9.3, 8 base pairs, then 12 base pairs are present. So next one, types of DNAs. So A type, B type, C type, D type and Z types. So generally DNA is a right-handed helix. So in only Z is a left-handed manner. So helix are coiling right-handed and left-handed condition. So these DNA, both strands of DNA coiled spirally. That means intertwined. Spiral in condition like ropes, then spears, strings of spears, etc. So due to this coiling, number of turns are present, number of coils, number of spirals are present. So each turn may contains the base pairs. So A DNA approximately contain 11 base pairs, then B DNA 10 base pairs, C DNA 9.3 base pairs, then D DNA 8 base pairs, then Z DNA 12 base pairs are present. Next one, each helix has some distance. So A DNA 25 angstroms, that means 2.5 nanometers, then B DNA 34 angstroms, then cDNA 30 angstroms, ddNA 24 angstroms, then zDNA 46 angstroms. Then the diameter, the width of ADNA is 26 angstroms, then BDNA 20 angstroms, cDNA 19, the unknown condition, then zDNA 18 angstroms. So ADNA is a widest, then zDNA is a thinnest. So ZDNA is a thinnest, then ADNA is a widest condition. Except BDNA, all are unstable condition. Unstable. So BDNA is a more stable, physiologically active condition. So BDNA is a high stable and physiologically active in a condition. Then in this DNA's repeating units are mononucleotide condition, but only in Z DNA dinucleotide condition. Dinucleotide condition. So in each helix, so 12 base pairs means 6 dimers are present. 6 dimers are present. So characteristic features of a bDNA. So DNA generally double helix in a condition, 2 strands are present. So the strand of the DNA not coiled upon itself. These are 2 strands are coiled upon itself. That means spirally coiled condition like a ropes. So due to this coiling form the number of turns, number of spirals, number of coils. So it is known as a one turn. So each turn may contain one small groove and one large groove. So it is nothing but large groove. That means major groove. That means a deep groove. So it is nothing but a smaller groove, shallow groove, minor groove. So one major groove and one minor groove is known as a one turn. So the major groove approximately 22 angstroms, minor groove approximately 12 angstroms. So total 34 angstroms, that means 344 nanometers. So each turn of a DNA has a 2 grooves. So minor groove, shallow groove approximately 12 angstroms, then major groove, deeper groove approximately 22 angstroms length. So 34 angstroms in nanometers, 3.4 nanometers. Then each turn contains base pairs. Each turn contains base pairs. So in bDNA approximately 10 base pairs are present. So one base pair means one purine and a pyrimidine. So for example, adenine and a thymine. In DNA, one standard. So one pyrimidine always comes opposite to one purine. Then vanillin and cytosine. So one strand of the purine always comes opposite to another strand pyrimidine. So purine and pyrimidine. In one strand pyrimidine come opposite to another strand purine. So this is known as a complementary nature. Adenine always base pairing with thymine, two hydrogen bonds. Then vanillin always base pairing with cytosine with three hydrogen bonds. So why always purines comes opposite to pyrimidines? Then pyrimidine comes opposite to purines. So why? Because generally purines are with a larger structure, bigger structure. So two rings of a structure. If a base pairing takes place between the two purines, so it is a big in a structure. So in middle narrow space is present. If two pyrimidines are pairing takes place, so it is a less smaller structure in condition in middle position. So large space is present. So not possible to maintain the uniform distance between the two strands of DNA. So because of this, so one big structure, that means purine always comes opposite to pyrimidine. Then one smaller structure, pyrimidine always comes opposite to purine. Adenine always base pair with thymine. Vanillin always base pair with cytosine with two hydrogen bonds and three hydrogen bonds. So in one strand of the DNA, purine always base pair with the pyrimidine. Then pyrimidine always base pair with the purine. One base pair means one purine and one pyrimidine. So these are complementary in a condition. So both strands are complementary condition. So run parallel but opposite direction. This is known as a anti-parallel condition. So each term has the 10 base pairs. That means 20 nucleotides are present. Then the distance between these two strands for 20 angstroms. That means 2 nanometers are present. 2 nanometers. In aDNA, largest wider. That means the diameter is present approximately 26 angstroms. That's why aDNA is a widest DNA. In zDNA, approximately 18 angstroms are present. So it is a thinnest in a condition. So characteristic features of bDNA. Next one, RNA. So RNA, ribonucleic acid. So RNA was the first genetic material. DNA evolved from RNA. So RNA essential for all important life activities. So in body of the organism, these important life activities evolved around the RNA. That means metabolism, splicing, translation, etc. So DNA evolved from RNA by some chemical modifications. So generally in DNA, deoxyribose sugar is present. In RNA, ribose sugar. In DNA, thymine is present. Then in RNA, uracil is present. Generally DNA is a double stranded condition, but RNA is a single stranded condition. These are two strands held by the hydrogen bonds. So these are three chemical changes may be taken in RNA to DNA. These are three chemical changes providing high stability to DNA compared to RNA. So RNA also acts as a genetic material in viruses. Some viruses. That means retroviruses. So generally RNA single stranded condition, but in some viruses it is a double stranded condition. So these are wound tumor virus, then rice dot virus, wound tumor virus, risa dot virus, double standard condition. Then RNA formation from DNA is known as a heterocatalysis. All types of RNAs, so transcription. So transcription means formation of RNA from the DNA. All types of RNAs. So transcription process. Generally in prokaryotes, transcription takes place in cytoplasm. In eukaryotes, RNA transcription takes place in a nucleus. Then generally 6 types of RNAs are present. So RNAs are 6 types are present: RNA, tRNA, mRNA, small cytoplasmic RNA, small nuclear RNA, and genetic RNA. These 3 are major RNAs. Then small cytoplasmic RNA, then small nuclear RNA play important role in splicing process. Play important role in splicing. Then genetic RNA, in some viruses, plants and animal viruses, RNA act as a genetic material. This RNA is known as a genetic RNA. Then first one coming to rRNA. So rRNA, R means ribosomal RNA. So ribosomes mainly constitute by proteins and rRNA. So rRNA mainly present in ribosomes. These are coiled around the proteins and ribosomes. That's why this is known as a rRNA, ribosomal RNA. It is a most abundant and highly stable RNA. Most abundant and highly stable RNA. Approximately 70 to 80% in cytoplasm of the cell. So generally RNA, all types of the RNAs are RNA, tRNA, mRNA formation for the transcription process in a nucleus, then comes out to cytoplasm of the cell. So these are freely present in a cytoplasm. So rRNA is the most abundant RNA, nearly 70 to 80% in total RNA. So it is a high stable RNAs. So generally ribosomes are 2 types. So these are 70S type of ribosomes, then 80S type of ribosomes. So in each ribosome 2 subunits are present. In 70S 50S, then 30S, in 80S 60S, and 40S type of subunits are present. Then each subunit contains some types of the rRNAs. rRNA generally 4 types are present. So based on the sedimentation coefficient in eukaryotes, 4 types of rRNAs are present. In eukaryotes, in eukaryotes 28S, then 18S, then 5.8S, then 5S. Based on sedimentation coefficient in eukaryotes, 4 types of rRNAs are present. So 28S, 18S, 5.8S, then 5S. Then in prokaryotes, so 23S, then 16S, then 5S rRNAs are present. Generally in prokaryotes only 1 polymerase enzyme is present, but in eukaryotes 3 polymerase enzymes are present: polymerase 1, polymerase 2, polymerase 3. So rRNA synthesized by the polymerase 1 enzyme. Then in eukaryotes 28S, 5.8S, then 5S is present in larger subunits. So 28S, then 5.8S, then 5S. These are 3 rRNA types are present in larger subunit. 60S. Then in smaller subunit 80S type of rRNA is present. In prokaryotes in larger subunit 23S, then 5S in smaller subunit S or RNA types are present. There, these rRNA contains a specific number of proteins. So, approximately more than 80 different types of proteins are present in rRNA. So, for example, in eukaryotes, in larger subunits, approximately 50 different types of proteins are present. In smaller subunit, 33 different types of proteins are present. In prokaryotes, in 50 yes, approximately 32 different types of proteins. In 13, 16 yes, 21 different types of proteins are present. In eukaryotes, in a smaller subunit, 18 yes, as a complementary sequence with mRNA kappa region. So these complementary sequence will be useful to recognize that particular sequence in mRNA cap, then bind, then promotion of translation process. In prokaryotes, so 16S in smaller subunit as a complementary sequence with mRNA kappa region. This 18S rRNA, 16S rRNA with proteins 21 and 33 different types of proteins will be useful to this smaller subunit of ribosomes binds with mRNA. So while the translation process. Next one, 5S rRNA. 5S rRNA provide the binding site for tRNA. Binding site for tRNA. So 18S rRNA, 16S rRNA in both eukaryotes and prokaryotes provide the binding site for mRNA. Next one, 5S rRNA in both prokaryotes and eukaryotes provide the binding site for tRNA. Next one, sedimentation coefficient of this rRNA is 45S in eukaryotes, then 30S in prokaryotes. So base pairs, so approximately are nucleotides, approximately 5000 nucleotides are present. Then molecular weight approximately 18 lakhs. So 18 lakhs of molecular weight is present. So main function of rRNA is provide the binding site for mRNA and tRNA, while the translation process ribosome RNA most abundant RNA and highly stable RNA. Next one, tRNA. So tRNA also known as a transfer RNA. The tRNA was first proposed by Greek scientist. That time he was called as a adapter molecule. Before proportion of genetic code, adapter molecule, transfer RNA. Why it is known as a transfer RNA? Why? Because it will bring the specific amino acids towards the ribosomes for translation process. That's why it is known as a tRNA, transfer RNA, adapter molecule. So more than 100 types of tRNAs are present. 20 different types of amino acids are participate in protein synthesis. For these 20 types of amino acids, 20 different types of tRNAs are present. Maybe 2 or 3 types of tRNAs bring the same amino acid. Next one, it approximately 10 to 15% in total mRNA. So most abundant is rRNA, 70 to 80%. It is 10 to 15% in total RNAs. Then tRNA also formation by transcription process. Generally rRNA synthesized at NOR region, nuclear organization region of chromosomes, NOR region. So transfer RNA. This transfer RNA is also known as a soluble RNA. Transfer RNA also known as a soluble RNA. Why? Because it is a easily degradable condition. Soluble RNA. So tRNA sedimentation coefficient approximately 3.8s or 4s sedimentation coefficient. Then base pairs approximately 73 to 85 base pairs are 90 or 93 nucleotides or base pairs are present. 73 to 85 or 93 nucleotides are present. Then molecular weight, so approximately 25,000 of molecular 25,000 daltons of molecular weight is present. Then tRNA shows the two-dimensional structure and a three-dimensional structure. So two-dimensional structure, so clover leaf model proposed by Holy scientist. Then three-dimensional structure, inverted L, inverted L, so proposed by Clark scientist. So two-dimensional clover leaf model, then three-dimensional inverted L condition proposed by Clark and Holy scientist. So generally tRNA has a 5 arms, that means 5 ends. So this end is known as a end, amino acid binding end. So next one, pseudo uridine, then dihydroxy uridine, then anticodon. Next one, variable arm. So tRNA has 5 arms. So amino acid arm, then DHU dihydrouridine, then pseudo uridine arm, variable arm, then anticodon arm. So after the transcription process in prokaryotes, there is no any processing. RNA becomes a mature RNA, mRNA, but in eukaryotes requires the number of processes. So primary transcripts to becomes to mature RNA, mRNA. So while the processing, so nucleotides are modified. So add the dihydrouridine, pseudo uridine, like that. Then amino acid binding site. Sir, here nucleotide sequence is CCA nucleotide sequence. This CCA nucleotides joins after the transcription process. So 3' end, at 3' end CCA nitrogen bases nucleotides are present. So 5' end is a rich of vanillin. That means G. So it is a binding site for amino acid. So generally tRNA single stranded condition at where foldings are present coiling, where only double stranded condition, that means base pairs are present. Next one, BHU loop. It is a mainly amino acid synthesis engine recognizing binding site. So amino acid synthesis engine binding and recognize site. Then pseudo uridine, it is a ribosomal binding site. Ribosomal binding site. Then anticodon loop. So in anticodon loop in tRNA nitrogen bases are complementary to mRNA. Complementary to mRNA. This tRNA will bring a specific amino acid, then joins to ribosomes. So while the translation process ribosomes binds to mRNA, by this anticodon complementary base pairs with mRNA, so joins add the amino acids to mRNA based on the complementary sequence of mRNA. This loop is known as a anticodon loop. So generally DNA is a double stranded condition. So two strands are present. So one strand polarity is a 3' end to 5' end, another strand polarity is a 5' end to 3' end. So these both strands do not carry the heredity information. So only one strand will carry the heredity information. So 3' end to 5' end, these strands act as a template strand. template strand. So this is also known as a antisense strand. So minus strand, non-coding strand, minus and non-coding strand. So these strand act as a template. This strand act as a template to formation of a RNA. So generally DNA regulates the metabolism and heredity information through the RNA. So DNA regulates the metabolism in organism's body through the RNA only. So in transcription process, the DNA strand with 3' end to 5' end act as a template strand. So this is known as antisense strand, non-coding strand, minus strand. Why this is known as antisense strand? So these nitrogen bases are complementary with this strand 5' end to 3' end. That means complementary to mRNA. So these nitrogen bases similar to tRNA. So anti-codon. In tRNA, nitrogen bases is known as a anti-sequence, like a anti-codons. So in tRNA, sequence is similar to antisense strand, complementary to mRNA strand. That means a codon strand. So in DNA, in this strand, the sequence is complementary to this strand. So this strand is known as a non-template strand. Non-template strand, then sense strand plus strand, then coding strand. This strand contains the codons. That means in this strand, the nitrogen base sequence is same to mRNA. That means RNA instead of thymine uracil is present. So remaining all sequence is similar condition. So 3 nitrogen bases code for a specific amino acid. This 3 adjacent nitrogen bases is known as a codon. So this strand contains the codons. Next one, mRNA. So mRNA is known as a messenger RNA. So why mRNA is known as a messenger RNA? Why? Because mRNA bring the all instructions from the DNA to polymerization or formation of a particular polypeptide. So mRNA bring the all instructions from DNA. These instructions coding in the form of nitrogen bases. So according to genetic code, triplet codon. So that's why this is known as a mRNA, messenger RNA. So mRNA approximately 3 to 5% in total RNA in cytoplasm of the cells. Then sedimentation coefficient of mRNA approximately 8 is present. Then nucleotides approximately 6,000 nucleotides or 6 to 10,000 or 12,000 nucleotides are present. Then molecular weight approximately 20 lakhs of molecular weight, 20 lakhs daltons of molecular weight is present. Then mRNA formation by the RNA polymerase 2. RNA polymerase 2 in eukaryotes. In prokaryotes only one RNA polymerase is present. Then in transcription process, the formed mRNA is known as a primary. In both the prokaryotes and eukaryotes, mRNA formation by the transcription process. So initially formed mRNA strand is known as a primary transcriptor. Nascent mRNA, hnRNA, heterogeneous nuclear RNA. In prokaryotes, there is no any requiring of processing. So this newly formed RNA to becomes the active mRNA. So why? Because in prokaryotes there is no non-coding sequences are present. All are the coding sequences. That means all are the functional sequences are present. But in eukaryotes, non-coding sequences, introns are present. So hRNA requires some processing to become active mRNA. These processes is known as a capping, then tailing, then splicing process. So capping, tailing and splicing. So by the capping add the one cap, then tailing add the tail. So in splicing remove the introns and joins the exons. So after processing, this hnRNA become to mature mRNA. So mature mRNA contain cap. Mature mRNA contain a cap. So while the capping add the one cap. So these cap contain 7-methyl guanosine triphosphate is present. So add the 7-methyl guanosine triphosphate. So while the capping. So these cap will be useful to binding of the ribosomes. Binding of ribosomes. Generally in rRNA. In eukaryotes, in the smaller subunit 80S RNA, then in prokaryotes 16S RNA as a complementary sequence with this cap. So by this complementary sequence, this RNAs recognize. So cap then comes and binds with the cap, then initiates the translation process. These capping also be useful to provide the binding site for ribosomes and provide the protection from cellular enzymes like nuclease enzymes, provide the protection from degradation. So after the cap, some non-coding sequences are present. This is known as a UTR region, untranslated region 1. Untranslated region 1. Then after this untranslated region, initiation codon is present. That means AUG code for methionine. After this initiation codon, so entire is a coding sequences are present. Then after the coding sequences here, termination codons are present. So these are UAG, UAG. Then UGA, termination codons. So after termination codons, untranslated region. So UTR2, non-coding sequence. These non-coding sequences will useful for effective translation process. So after this UTR2, so tail is present. So tail. So while the tailing, processing of tailing, add the one poly A tail. So poly A tail, add the 200 to 300 adenylate residues. So this tail will be useful for disassociate ribosomes. So after completion of translation processor, and also provide the protection from cellular enzymes, degradation from nuclease, etc. This is known as a mature mRNA. Mature mRNA. So mRNA bring the all instructions from DNA for the polymerization of particular polypeptide. That means the formation of proteins. So this contains all sequences for formation of particular polypeptide. Then the lifetime of mRNA is a very less condition. So few seconds to few hours in lower forms of organisms. It may be several days in higher organisms. Then generally mRNA is two types. So it is a monocystonic condition, then polycystonic condition. Monocystonic and polycystonic. So monocystonic generally present in eukaryotes. Then prokaryotes. Eukaryotes Monocytonic means it will form the only one type of polypeptide. Polycystonic means form the more number of different polypeptides. So polycystonic in prokaryotes, monocystonic in eukaryotes. This monocystonic condition also known as a split gene. Is also known as a split gene. So why? Because more number of unfunctional. So codons sequences are present in eukaryotic DNA. So codon sequences interrupted by non-codon sequences. Is known as exons. Codon sequences is known as exons. Through the splicing process, removal of these introns, then joining the exons. This is known as a splicing process.